Hi students, a little recap on our lesson from Thursday, which we kind of ran out of time because the experiment went a little bit over time, and a few tech issues as well with our presentations, but that's okay. We can catch up. I'm making this for you to watch before our next lesson on Monday, so please make sure you do. It's great. You can fast forward, you can rewind, pause, play again, etc. So just a little reminder to read pages 192, 194. This was homework from last week. Respond to Qs 1-4 in your science books. Please write all your responses into your science book, not onto Teams. Just a little reminder about that. Okay, let's have a look. I've sent a couple of emails to those that did not get their formative assessment in on time. Make sure you follow that up. So today, the chemical structure of DNA, syllabus learning objectives. You can pause here and read that. You don't have to listen to me read it out. So please pause and have a read yourself. And a little review. So this should be from year 9, year 8 science, looking at the difference between prokaryotic versus eukaryotic cells and the DNA within them and how that DNA is contained. So prokaryotic DNA, you can read this yourself. It's found freely in the cytoplasm. And just a little reminder, prokaryotic organisms are not you and they're not me. They are simple organisms like bacteria or archaea. Eukaryotes are complex organisms like us, like animals, plants, etc. So prokaryotic DNA is found freely in the cytoplasm. And a little fun fact, pro meaning before and karyotic nucleus. So this is an organism that does not have a nucleus. So generally, there's always exceptions to the rules. So prokaryotic DNA is found freely in the cytoplasm, is circular in shape, and may contain extra chromosomal plasmids. Eukaryotic DNA is contained within a nucleus. So that is one of the main differences between the DNA of the prokaryotes versus eukaryotes. We have a nucleus where our DNA is bound, and a prokaryotic organism does not. Our eukaryotic DNA is bound to histone proteins. We have a large amount of non-coding and repetitive DNA, and they are linear in shape. So what I'd like us to look at today is DNA replications. It's quite a complex process, but when you break it down, it is very simple and it makes sense. So all cells, as you know, are derived from pre-existing cells. And before a cell can divide, so think mitosis and meiosis, which we'll be looking at soon, the amount of DNA within that cell needs to be increased. And this is where replication comes in. It's the process where one DNA molecule is duplicated to make two identical DNA molecules so that the next generation of cells can contain the exact same genetic information. So remind yourself of the structure of DNA. It's a double helical strand, or double-stranded, and these strands run anti-parallel. That means that one strand runs from the 3' to the 5', and the other strand runs from the 5' to the 3'. So there's a great video here that I want you to pause now and go and watch. If this doesn't work for you in the presentation that's on Teams, you can simply just Google DNA replication, amoeba sisters, and this will come up. I love the animations they have. It's really memorable, and it's definitely suitable for a Year 12 knowledge range. So please pause now, go and find this video, watch it, and come back. So you are back. I hope you enjoyed that little video, and hopefully you did make a little bit of notes related to some of these words right here: helicase, 3', 5', etc., replication fork, etc. So given that you've just watched that video, the rest of this presentation will make perfect sense. Okay, so this is a simplified version of replication in action. This point right here is what we call a DNA replication fork. And as the DNA unzips, that fork obviously moves along. We have our leading strand running from the 3' to the 5', and our lagging strand running from the 5' to the 3'. We have replication occurring in the leading strand in a continuous way, and replication occurring in the lagging strand in a discontinuous way. Now let's go into this just a little bit further. by looking at this diagram here. Now, I didn't make this diagram. This is not mine, but my explanation will hopefully make this a whole lot easier for you to understand. So before I go into it, just have a look at this DNA strand. We have it running from a 3 to a 5 on this side, the leading strand, and a 5' to a 3' direction on the lagging strand. Okay, so first of all, in the beginning, we have an enzyme called DNA helicase. And this enzyme, now you can generally determine if something is an enzyme or not if it ends in ASE, where a lot of these indicators are and primers are that are involved in DNA replication. So DNA helicase unwinds and separates the strands by breaking the weak hydrogen bonds of the complementary base pairs. So those bonds between the adenine and thymine and the guanine and the cytosine, they're broken by DNA helicase. So DNA helicase helps to unwind that double helix strand. And next, the enzyme DNA polymerase adds complementary DNA nucleotides. So let's have a look at this in a little bit more detail. So we'll start up here on the leading strand. So as I said, DNA polymerase can only add nucleotides because of the anti-parallel nature of DNA. It can only add nucleotides in the 5' to 3' direction. So that means that they get added in this direction here. From this direction here. I need my little marker here. So we have a 3' on the leading strand, and DNA polymerase can only add nucleotides in the 5 to 3 direction. So it can start up here and replicate easily and continuously this way. So here we have the DNA polymerase just cruising right along this template strand, adding individual nucleotides. So you'd have your adenine and your thymine and whatnot. It'd be the complementary bases to those would be added beautifully along here. It's continuous. So in order for DNA polymerase to know where to start, there would have been a moment or a place where RNA primer would have been added, and it gets added by the enzyme RNA primase. So as soon as that RNA primer is added, maybe up here, DNA polymerase now has a binding site where it can attach to that leading strand of DNA and then add nucleotides in that 5 to 3 direction. So if you have a look at the lagging strand down here, you'll notice that's just a little bit different. There's a little fragmented, oh my goodness, brain work, fragments appearing along that strand in a discontinuous way. So the lagging strand, so we go to the replication fork, as those strands are unzipped, you can see that lagging strand is in a 5 to 3 direction. Now that's the same direction that DNA polymerase must add those complementary bases. So it can't go in that same way. It has to go in this way, in this direction. So what happens is, as that the template strand, let me just get my little marker. Where is it? Just a sec, I'll grab it. Okay, here we go. So as the DNA gets unzipped, the template strand is actually traveling in the 5 to 3 direction. Polymerase has to go in the 5 to 3 direction, which is this way. Okay, so as that is the only way it can go in, the lagging strand ends up being completed discontinuously as compared to the leading strand, all right, which is completed continuously. So these fragments that are being completed are known as Okazaki fragments, and that's simply named after the Japanese scientist, who discovered them and discovered some other elements of replication. So in order to complete replication of the lagging strand, the Okazaki fragments need to be joined together. So you can't have a DNA strand being made with gaps in between. So those gaps need to be joined together, and this is done by another important enzyme called DNA ligase. So I hope that makes it just a little bit easier to see how replication occurs in DNA. Once you realize the difference between the leading and the lagging strand, and get to know the names of the enzymes involved in the process, it's a lot clearer. Okay, so prepare to have your mind blown. Oh, I just need to get rid of my pen. Okay, so I have this amazing video here. Now, let me preface this by saying that you don't need to know all of this. It's just super interesting at what science has discovered and technology allows us to see. So please click on this link in the presentation. I'm going to please pause our presentation here and go into the PowerPoint on Teams if you haven't already got it open on the side, which you should have. But it's also in the Wakelet as well. It's absolutely fantastic. Please go and have a look. It's not very long. It's about 4 or 5 minutes, and come back once you're done. Okay, so looking a little bit closer at eukaryotic DNA. So as you know by now, the average human cell contains almost 2 meters of DNA. So cells are so tiny. How do we fit 2 meters of DNA into 2 micrometers of space? Well, it's quite amazing, really. So to organize that DNA and stop it tangling prior to duplication or replication and separation, each DNA strand is wound at intervals around 8 proteins called histones, and these form a DNA histone complex called a nucleosome. Okay. So here's a little bit of a closer look at that. So those 8 histones have chemical tails exposed, which can be chemically modified by binding with epigenetic factors. So this locks away the DNA so it can't be copied to make a protein, or they can be freed and drawn into the histone protein if DNA needs to be copied or to make a protein. So there's 2 ways it can go, and the histones help with that. So how do these histones help in that process? So if we think about those 8 histones forming a group, it's actually 4 but doubled. So we have H2A, H2B, H3H4, and then H2A, H2B, H3, and H4. So these colors are really useful. So basically there's one H2A, another H2A, one H2B, another H2B, and so on. H3, H3, H4, H4. And those guys just get all together and form a group. And we have them over here on this side here. We add DNA into this scenario. And what happens is that DNA wraps itself around that histone complex twice, or 1.75 times, 1.75 times, and that helps form that chromatome. And this stops the tangling of those long threads of DNA so that replication or protein making can occur without error or with ease. So as that coiling continues, I'm just going to get rid of my pen, that coiling of the DNA occurs. And this is what you may be more familiar with. So we have our DNA double helix. We have those 8 histones together coiling, and the DNA coiling around those histones. And as they coil tighter and tighter and tighter, they become supercoils. And as they get extremely tight, they form this beautiful shape that we are very familiar with, known as a chromosome. We're going to dive more into chromosomes next lesson, and then which leads into meiosis and mitosis. So feel free to read ahead on any of those topics. Your homework for this week is to read these pages in your text and complete 4 questions from page 197. If you wanted to review any of that DNA lab work that we did, there's some links in the presentation. 4 of you have submitted at this point in time your assessment on Teams. Thank you so much, and I've already provided feedback. All of this other work, those pages from this and last lesson and questions, are due on Monday the 9th. I wish you all a fabulous weekend. Take care.